everyone. Welcome to another episode of Inside the Sciences. I am Sheena Burhal and I am your host. And I'm actually quite excited with today's episode because we're going to talk about a topic that is very close to my heart as a microbiologist. We're going to talk about microbial cell culture and the importance of movement in these types of cultures. So in the world of microbial cell culture, movement isn't just about mixing. It's also about survival. Some microbial cells, they demand motion. They would only thrive when the liquid around them stays in rhythm. While others, they would rather just avoid the motion altogether. So the question now is, how do scientists know when to hit start on an orbital shaker? And what happens when the wrong cells get caught in the mix? So we're going to break it down in this episode. And we are joined by Tahani Vidraputri, a product management executive here at Esco. She will talk about why some microbial cells need motion and others don't. And we'll also dive into the role of orbital shakers and why they matter and when it is crucial when getting them so that you can have better results for your cultures. So Tahani, thank you very much for joining us in today's episode. Okay, we're gonna talk about microbial cultures and the importance of movement particularly. And probably we can start with the basics, when we're talking about microbial cell cultures, there are microbial cells that they don't need to be shaken, like they don't need motion, while others, there are microbial cells that they need motion. So can you tell us the difference between the two? How do they differ?
Yes. So not all microbes need shaking because some of them keep floating in the liquid and some of them will attach to the surface. Like planktonic microbes, for example, like Escherichia coli, they will keep floating in the liquid and shaking can help them to improve the oxygen transfer rate there and will make the uniform distributions for the nutrient, while for the surface attach or biofilm forming microbes, they will attach to the surface or they will produce an extracellular matrix. And I think shaking unnecessary for them and it will disruptive for them. So I think the needed of the shaking, it depends on their growth natural mode.
Okay, so I think it's because they have different growth strategies. Yes. All right. Okay, so for someone new to microbial cell culture, say a microbiology student or a laboratory staff who is doing microbial cell culture and they're new to this kind of methods, how can they figure out if the cells that they're working on needs shaking or don't?
The simplest way is to observe them in their early growth stage. Like if the microbes keep spread out in the liquid or they make the medium cloudy, they're likely a planktonic microbes. But if they attach to the surface and then they make a layer or clumps, they're likely surface attached microbes. And I think that's all for the
okay. So you take note of those things, especially if you're doing microbial cell culture. Now, how about when the cells that they don't actually need to be shaken, but accidentally they got shaken? What would happen? Will it cause damage?
Yes, of course. Yeah, it can damage the cells. It can change the structure of the cells, such as in HEVA, in filamentous bacteria. Shaking can break the biofilms there before they mature. It also can make the cells stress. Also can change the genes of expressions. And also, they can reduce the cells densities. So it will change the results also. We cannot meet our expectations also.
So it's really important to check if your cells need shaking or not. Okay, so based on your experience, what are how do the benefits of orbital shaking translate into better growth, of the culture and consistency, like for example, nutrient and oxygen distribution?
Shaking can improve the oxygen transfer rate from air to liquid, and they can make a uniform distributions of the nutrient, can prevent from for the cells from settling at the bottom.
Mm-hmm.
And for especially for the aerobic planktonic microbes, I think they shaking can make them grow faster. Also, can make them has higher final cell densities.
Okay.
So yeah, and the best things is they will have the optimal or consistent replications.
Yes.
Because they have or experience the same conditions there. So yeah.
Okay. How about what's the impact of like a lack of movement in cell cultures? Like, are there any visible signs that the culture wasn't, incubated with the right kind of motion?
Yeah, without proper movement, the cells will be settling at the bottom, and then it will reduce the cell densities also, and make their growth slower. So yeah, it will affect the quality also of the cultures.
Okay. And how does shaking really impact the outcome, particularly of suspension cultures in particular? Why do these cells need that constant movement?
Yeah, like I've mentioned before that this can make an optimal or unstable conditions for the cultures. So they can get the same conditions there, not too much or not too less for the nutrients and oxygenations, and prevents them from settling at the bottom. So the quality will be uniform also and same.
Okay, so now that we understand how some microbial cells need motion, others don't, and we also understand the science behind orbital shaking, the next question is how do we ensure that that our shaking is done right? Because you can have the best shaker incubator, but still it can fail to deliver consistent results if it's not set up properly, right? So how do we make sure that shaking is actually done right? So I guess my first question to you for this one is like if you're working with if you're using a shaker incubator, what settings matter the most and why?
I think the two important things is the speed and the temperature, because they lead directly to the metabolism of the cells, also the microbial growth. So I think we should set the speed, so set the RPM, and we can get the stable conditions for the microbes. Yeah. And I think have a stable temperature also can lead to the microbial growth also. So I think having shakers that has built-in refrigeration, that will be a great things, because it can also use for our sensitive samples also, because we have that refrigeration things in our shaker.
Okay, that's actually interesting. How about how can someone know if their shaker is running correctly or if something is wrong?
Okay, so usually I check two things. I check the uniformity of the movements, and I also check the stability of the set conditions. So if we heard or if we see the vibration that wasn't there before, I believe that is something off. And and I think having a large display, like 10-inch interface, that will also help us to see or monitoring our set conditions, like the temperature, the speed, and if this not align or match with the actual growth pattern, I think it's a red flag.
Okay, so in your experience, what are some of the common mistakes that you've seen people make, especially when they're using orbital shakers?
Usually people will overloading the capacity, like push all of the things, and it will disturb the airflow there, and will reduce the oxygen transfer rate there. So it will affect that. And also I usually see that people not clean their shakers, not clean the chamber, not clean the platform between the use, and it's caused the contamination risk. So I think having dual UV sterilization for our shaker is the best things, because we don't need to manual cleaning the platform, and it will reduce the contamination risk, right?
Actually, very practical. How about for laboratories? that are thinking of upgrading or they want to buy a new shaker, what are the things that they should look for?
From my side, I think the 3 points that need to be considered is the stability, the capacity, and the flexibility. So we should see that can this shaker set the temperature stable and precise? Mm-hmm. Is it big enough for our samples in now or in the future? Or can this shaker hold our samples, our large and small samples? Mm-hmm. And I think for having the stackable design would be great because we can save the space in the labs, and but we still can hold a lot for the samples.
Yes, I agree. Should be stackable. Mm-hmm. Okay, so and when we're talking about orbital shakers and microbial cell cultures, we know how important microbes are, particularly in different industries. And in different industries, they have laboratories. So maybe we can talk about now the practical, the real world applications of shaking our microbial cell culture. So in your orbital shaking, it's often associated with microbial cell cultures, but beyond the laboratories are also practical applications. So can you talk about how shaking is used in fermentation or food production, for example?
Yeah, in fermentation, like making soy sauce or making yogurt, I think, shaker is needed because, like what I've mentioned before, that this will be make a uniform distribution of the nutrient and oxygenations there. Mm-hmm. Without the shaking, it will lead you to it will affect the taste and also the results, the quality also. And luckily, our shaker has 3D triple eccentric drive system there, so we can have heavy loads of fermentations maybe, but we no need to worry because it will not overheating there. Yeah. So it will not reduce the oxygen transfer rate there also. So we will have the uniform results batch after batch.
A quality result, particularly in fermentation, because there are a lot of food products that are fermented, right? And they're very important in the food industry. Now, how about for, let's say, vaccine production? Can you explain how shaking impacts products like vaccines that are grown particularly, say, in bioreactors? How does this technology support large scale production?
Yes, for probiotics, shaking can prevent sedimentations, while in vaccines, it's more into the stable conditions, optimal conditions, so the cells can form the antigens, the right antigens. And in large scale, we can use our stackable design here because we can save the space and also can have a lot of capacity. But we can have the same footprint there. So I think it's a good thing.
Yeah, so really in based from what you're saying, our Esco stackable incubator shaker is really very practical. Right? Okay. So but aside from the food industry, aside from vaccine production, what other industries are benefiting from shaking technology, and how is it helping them scale their operations?
Like cosmetic manufacturing also use that things. Biofertilizer and bioremediation manufacturing also use that things. So having for our incubator stackable shaker, we have brushless motor and low noise operations. It helps them to have a quieter lab and also less energy waste. So it will be more sustained. Yes.
Green technology.
Yes, correct.
Yeah. Okay. So, you are experienced in this field. You have a lot of experience in this field, and I want to get your opinion for my last question to you for this particular episode. Where do you see the future of shaking technology heading? Are there any new innovations or applications on the horizon that you can see?
I think in the future we will have the smarter shaker, maybe. Like
That's interesting.
it will be like maybe in the future, I don't know when, but I think we can make this like AI based or something to setting or monitoring, and we will have the real time analytics or maybe like remote monitoring. And I think our Esco stackable shaker already takes step for that. Like we already have 10-inch touchscreen, and we have data tracking, and also 912 days of data storage that can be exported via USB. So I think, yeah, hopefully we can develop all of the.
It's good that Esco actually has those innovations. All right. So thank you very much, Tahani, for joining us. Very, very insightful, information coming from you. Thank you so much for being with us. Okay. All right. So innovation in science is not always about new discoveries. Sometimes it's also refining the basics. And sometimes as simple as consistent shaking can make all the difference. So whether you're just starting on an early stage research or you want to scale up your laboratory, it's important to understand how movement impacts cell cultures, and it can actually give you better, more reliable outcomes. If you want to know more about laboratory incubation technology and smart solutions in laboratory, visit www.escolifesciences.com. Once again, I'm Sheena, and this has been Inside the Sciences. Bye!